I want to say is welcome to the third seminar of this seminar series, and a big thank you to our two speakers for coming up, jumping in at such a short notice. So I let the introduction to you for Julia and Anastasis, and looking forward to the talk.
Great. Well, thank you, Teresa, for also coordinate the whole thing, because it was a last minute almost disaster, but hopefully a great success. Thank you all for being here or on YouTube. And I should say that there was a last minute, as you understood or saw in the email, change of plans. We initially invited Benoit Morel to present his work on tree reconciliation methods, but unfortunately something happened to him and he couldn't be here. So we wish him all the best, and we do hope to have him on a future seminar. But we are very excited and very grateful at the same time that we have Julia and Anastasis joining this seminar. And so just to mention a bit of their background, they're both computer scientists. Julia has a master's from the Karlsruhe Institute of Technology, and she has started her PhD in the Exelixis lab, Alexis Stamatakis lab in 2022. So her talk will be on Pythia, a software that tries to predict the difficulty of phylogenetic analysis. And then we will have a follow-up talk by Anastasis Togkousidis. He has a diploma in electrical and computer engineering from the Aristotle University of Thessaloniki in Greece. He also started his PhD a couple of years ago in Alexis' lab. And his work is building upon Julia's work, and he uses the predictions of Pythia and has worked on adapting RAxML in accelerating phylogenetic inference under maximum likelihood using dataset difficulty. So the format of the seminar is that Julia will give us a talk first and a very short demo, and then the same will happen with Anastasis. And then we will have time for questions to both of the speakers. So Julia, if you're ready, we are really excited to hear your talk.
So let me share my screen. I hope you can see my slides.
Yeah.
Perfect. So hi, everyone. I'm Julia. Thank you for the invitation and the opportunity to present my research here. And my talk will be about predicting the difficulty of a phylogenetic analysis. So before we dive into the topic, let's cover some basics. Phylogenetic analysis includes the reconstruction of the phylogenetic tree. So we have sequence data of organisms that we want to study. And for these sequences, we want to know the evolutionary history. So we compute a phylogenetic tree. Ideally, we want to have the true tree, so the tree that correctly shows us what happened during evolution. But unfortunately, as you might know, this problem is not so easy. With the number of sequences in our data increasing, the number of possible tree topologies increases over exponentially. So we cannot simply define an accuracy metric and apply it to all possible tree topologies to select the best one. Instead, we have to rely on some kind of tree search heuristic. And over the course of time, many such methods were developed, and coming up with better and faster methods is still a very active field of research. But in general, when we want to infer phylogenetic trees, we do have to do some kind of trade-off between speed and accuracy. So we can use fast methods such as the maximum parsimony-based ones or neighbor joining, but they are not as accurate. There are more accurate ones, such as the maximum likelihood method or Bayesian methods, but these methods are very slow. So for example, when we're inferring trees with the maximum likelihood method, the jobs can take hours or even days to finish, especially for alignments with many sites or taxa. Now we could say, well, that just comes with the job, and we just start analyzing the data with such an accurate method and we wait. But with data sequencing becoming faster and cheaper, we somehow need to better select what data we want to analyze and how we want to analyze it to obtain useful insights. Now, with all this in mind, let's talk about the difficulty in this context. And to do so, let's look at a typical analysis scenario. So we have a multiple sequence alignment for the sequences that we want to study, that we want to find the phylogenetic tree for. So we run our favorite tree inference method, for example, maximum likelihood, and we obtain a bunch of trees. Now, of course, we don't stop the analysis here, but we do additional post-processing like bootstrapping, or we apply sequence tests. And hopefully all these analyses will point towards a single or maybe a few very similar, well-supported tree topologies. Unfortunately, this is not always the case. Sometimes all these analyses lead to multiple highly distinct topologies that are all equally well supported. So the difficulty in this context describes exactly this behavior of an MSA. So it describes, so to say, the ruggedness of the tree space. And an easy MSA yields one or few very similar tree topologies, so we can be fairly certain that we uncovered the evolutionary history for our set of species. Whereas a difficult MSA yields multiple distinct yet statistically indistinguishable topologies that tell contradicting stories about how the species evolved, and we have no idea which one is actually the correct one. In the case of maximum likelihood analysis, we would say an easy MSA has a single likelihood peak, and with a difficult MSA, we observe multiple likelihood peaks. Now, the problem with this typical analysis scenario that we just talked about is that we only know how the MSA behaves after performing many tree inferences and post-analysis. And as I said, methods like maximum likelihood can be very slow. So the feedback loop for this is very slow. And if we find out that our MSA is in fact difficult, we might have wasted an enormous amount of time and resources for an analysis that won't even lead to great conclusions or even conclusions at all. So at this point, I hope you all agree that it would be nice to get a feeling of the behavior of an MSA, so the difficulty of an MSA before we do all this time and resource intensive analysis. And this is exactly what Pythia does. So Pythia is a difficulty prediction tool based on machine learning. And to train Pythia, we pose the problem of difficulty prediction as a supervised regression task. That means that we train the machine learning algorithm to predict the difficulty using ground truth labels as target. And the difficulty is a value between 0 and 1, with higher values corresponding to more difficult MSAs. Now, one main issue we had during our research was how do we quantify the ground truth difficulty for our labels for training? I will skip the details here to leave some more time for a small demo later on, but it's basically based on an intensive tree inference using 100 maximum likelihood trees. Now, the current version of Pythia is trained on about 12,500 empirical MSAs, including DNA, protein, and morphological data. So we cover basically all kinds of data that biologists might want to analyze. And in the current version, the mean absolute percentage error is 1.7%. So on an independent test set after training, Pythia predicts the difficulty wrong on average for 1.7%. Or the prediction is off by on average 1.7%. So our predictor is actually very, very accurate in predicting the difficulty. Now let's talk a bit about the features that we use for the prediction. Now, the ultimate goal is to save runtime. So the prediction and the computation of the required features should be fast. We experimented with different possible feature sets, and we ended up using this subset of 10 features that are all fast to compute, as I will show you on the next slide. 5 of these features are attributes of the MSA. So we use the sites over taxa ratio that relates the length of the sequences to the number of sequences in the MSA. We also use the patterns over taxa and patterns over site ratio that relates the number of unique site patterns to the number of sequences and the length of the sequences. And we include the percentage of gaps and invariant sites. Invariant sites means that the site never changed during evolution, which shows as a identical state throughout all taxa in the MSA. So the site is identical for all taxa. And we have 3 features that quantify the information content of the MSA. We use the Shannon entropy that we compute per site and then average it over all sites. And the other 2 features are based on the number of patterns and the frequency of each pattern in the MSA. Finally, we have 2 features that are based on a fast but less accurate tree inference heuristic using maximum parsimony. And for these 2 features, we first infer 100 maximum parsimony trees, which compared to a single maximum likelihood tree inference is very fast. And we then use the fraction of unique topologies among these 100 trees, and as well as the average topological distance between the 100 trees as features. And I didn't show you the definition of the ground truth difficulty that we used, but these two features are actually very related to this quantification. So they give kind of a sense, a very fast estimate of how the tree space actually behaves. Now let's have a look at the runtime. So this figure shows the runtime of feature computation for an MSA relative to a single maximum likelihood tree inference with RAxML-NG for the same MSA. And the x-axis here shows the size of the MSA as number of taxa times number of sites. And the y-axis shows the relative runtime in percent. And this red horizontal line here depicts 100%. So everything above this line actually exceeds the runtime of a single maximum likelihood tree inference. But the vast majority of MSAs, and especially larger MSAs, are actually, it's way faster to compute the prediction features compared to a single maximum likelihood tree inference. And if we remember that without Pythia, we would have to perform multiple tree inferences and post-analysis to even get a sense of the difficulty. So we can see that using Pythia can actually bring great value because it allows us to get an insight into the behavior of our phylogenetic analysis before having all these time-intensive phylogenetic inferences and post-analysis. Okay, so given I've convinced you that it makes sense to use Pythia, how can you actually use it? So the easiest way is to use the command line interface, and that's part of a Python package called Pythia, and you can get it from GitHub or install it via Conda. Pythia also includes a Python library such that you can use all the functionality of Pythia that you can use in the command line from code in Python. There's also a C library for Pythia. This is more complicated to set up, so we won't talk about this here, and we will focus on the easy-to-use command line interface. And before I switch over to my terminal for a quick demonstration of the command line interface, just 2 quick notes. So you can input your MSA in either phylib or FASTA format, and Pythia works for DNA, protein, and morphological data, so you can basically analyze any kind of phylogenetic data that you have as long as it's an MSA. So let me switch to the command line. I hope you can now see my command line or my terminal. Okay, I'm getting a thumbs up, thanks. So we can start by getting a help message for Pythia. And this help message basically just tells you what the options of Pythia are and what kind of analysis you can make. And the most important things are you need to provide Pythia, of course, with an MSA. So you need to provide a path to the MSA that you want to analyze. And you need to provide a path to RAxML-NG. Pythia uses RAxML-NG internally to compute the parsimony trees. So let's run a quick example. So as you can see here, I provided Pythia with the path to RAxML-NG on my laptop and to an example MSA that I ship with Pythia. So it's a quite fast to compute example, as we will see in a second. We will also use here for this demo the verbose mode, which means that Pythia will print the values of the prediction features. So we can kind of make sense why Pythia predicts the difficulty the way it does. And we will also use the benchmark mode to get a benchmark how fast Pythia computes the features. And finally, we will use something called the Shapley values, which I will actually talk about in a second when I'm back to my slides. So let's run this. And for every execution of Pythia in the command line, you will get this kind of status update to see what Pythia is currently working on. And finally, we will get the difficulty for our MSA as 0.16. And Pythia says that this is a very easy MSA. And in fact, this MSA is known to be easy with a single likelihood peak based on extensive maximum likelihood tree inferences. So apparently for this MSA, Pythia did the right thing and predicted it to be very easy. Since we use the verbose mode, we can have a look here at the features that Pythia used for its prediction. And so we can kind of make sense or try to make more sense of why the prediction is easy. Now we can see that the signal in the data is pretty good. We have lots of sites in relation to the number of taxa and the same for the number of patterns. And if we look at the relative topological distance between the maximum parsimony trees, we can see that they differ only at about 20%. So even the maximum parsimony tree inference kind of looks like it converged to a few very similar tree topologies. Now we also asked Pythia to print the runtime summaries. That's the -B option up here. And the total runtime for this prediction was about 8.5 seconds. And if you compare it to a maximum likelihood tree inference for this data set, that actually takes 16 minutes. So we do have quite a nice speed up here to get some first sense of this of this data set and its behavior under phylogenetic analysis. Now I also used here this --shep option. And what Pythia does when you set this option, it will print a so-called waterfall plot of Shapley values. And I will explain what that means when I get back to my slides. So let me switch back to the slides. Okay. So this is what the waterfall plot shows. So the Shapley values are one method of explainable machine learning. And the idea is to visualize what feature contributed how much for one particular prediction. So in our case, which feature contributed how much to the overall predicted difficulty. And it's very important to know that these Shapley values here are only meaningful for this one example, and we cannot really generalize to other examples. But let me explain to you what you can see in this plot. So at the top here, you can see Pythia's final prediction, which is, again, as we've seen in the command line, 0.16. So it's an easy MSA. And what you can see in the bottom here is Pythia's baseline. So during training, Pythia learned that it should start at a difficulty of 0.35 and then adjust the difficulty based on the computed feature values. And this adjustment you can see here with these the red bars pointing to the right mean that this feature contributed towards the MSA being more difficult, and the blue bars pointing to the left means that this feature contributed to the MSA being classified as more easy. And the further up a feature is in this list, the more it contributed to the overall prediction. And in this case, the feature with an astonishing impact on the prediction is actually the patterns per taxa ratio. And as we've already seen in the command line with the verbose mode, this data set has a very good signal. So we would expect that we would get like a very conclusive analysis. And apparently Pythia seems to agree that this pattern per taxa ratio is important for this one, and it's really good. So it actually decreases the difficulty from the baseline by quite some margin. Now, as I said, these values are only meaningful for this particular MSA, so for this particular set of features. And the Shapley values do not mean that removing this pattern per taxa ratio would result in a prediction that is 0.23 more difficult. It also does not mean that for another data set with a patterns per taxa ratio of 155, the difficulty will decrease by 0.23. So if you want to make use of these Shapley values to kind of get an insight into your prediction for your MSA, make sure to read a little bit more about what Shapley values can say and what they can't say. So to summarize this little demo, I've shown you the Pythia analysis for an example MSA that is known to have a single likelihood peak, so it's expected to be easy, and indeed Pythia identifies it to be easy. The runtime on my laptop was, okay, I said here 10 seconds, but it was actually only 8.5 seconds. And in contrast, a single maximum likelihood tree inference takes about 16 minutes. You might say now, okay, 16 minutes is not that much. And easy means that we would have detected this after a few tree inferences already. So let me show you another example where the runtime gain is way more important. And this is actually for an MSA of COVID data that my colleagues analyzed 2 years ago. And one conclusion of their publication was that the COVID data has a rugged likelihood surface and is very difficult to analyze. So let's have a look at what Pythia has to say about this. Well, according to Pythia, the difficulty is 0.82. So it's basically a hopeless MSA. It's very, very difficult. And looking at the features, this kind of makes sense. So if we look at the sites per taxa ratio, it only has about 6 sites per taxa, which is not that much signal. And the parsimony trees, they don't really reach like a conclusive tree topology. They have an average topological distance of 80%. And this entire prediction took about 12 minutes on my laptop. And in contrast, inferring a single tree with RAxML-NG takes more than 12 hours on a way more powerful machine. And of course, for such a difficult MSA, we would need to infer many, many more than just a single tree. So this is an example for an MSA where one really should decide whether the analysis will lead to a helpful insight or if it's maybe just a waste of resources that won't lead to any conclusion or insights, as we cannot seem to recover the evolutionary history for this dataset. Which leads me to the use and misuse cases of Pythia. Now, I encourage you to predict the difficulty prior to any phylogenetic analysis. Using this predicted difficulty, you can make an informed decision about the appropriate setup for your MSA. If your MSA is easy, you can maybe start with a few tree inferences, confirm that the tree search behaves as you would expect and shows a clear likelihood peak. If your data set is a bit more difficult, you might want to perform additional statistical tests and then summarize all tests that are indistinguishable from the set of inferred trees. Now, it's very important to mention that the difficulty does not predict the number of tree inferences required to sufficiently sample the tree space of your MSA, because this number also depends on what kind of tree search heuristic you're using. So if you're using IQ-tree, you might need a different number of tree inferences compared to RAxML-NG, simply because RAxML-NG and IQ-tree implement different search heuristics. Now, if your MSA is predicted to be very difficult, you should consider adjusting your data if you can, because the analysis will most likely be very uninformative. You can do so, for example, by adding more sequence data to each taxon if you have the option to do so, so that the signal and information content increases, or you might consider removing some taxa from the data to shrink the tree space of possible topologies. And of course, you can consider an adaptive search heuristic based on the difficulty, which luckily you don't have to think about because Anastasis did all the work, as you will hear about in the next talk. And before I end this short talk, let's quickly summarize what I've shown you. So I presented Pythia, a difficulty predictor that predicts the ruggedness of tree space under phylogenetic analysis. Pythia can predict the difficulty prior to time-intensive tree inferences. It's accurate and fast. It's in most cases way faster than a single maximum likelihood tree inference, especially for larger MSAs. And if you want to know more about Pythia, the paper with more details is available in the Molecular Biology and Evolution journal. And of course, I'm happy to discuss any questions or suggestions that you might have. And with this, I thank you for your attention and I hand over to Anastasis.
Thank you very much, Julia. That was very clear and great. I mean, I look forward to actually trying it.
Nice.
So Anastasis, if you're ready. And then we can...
There is one question on the chat. I don't know whether we shall answer it now or...
I think we can do all the questions in the end because the two topics are very close together, relevant.
Okay.
So, yeah.
Makes sense then. Yeah, so I shall share my screen. Okay. Yeah. So I'm sorry, I have to find the tab now. Yeah, so that's the tab. And yeah, now I think you can see my screen.
Yes.
Yeah, but I have to go back. Okay, so yeah, I'm Anastasis Togkousidis and I will present the adaptive version of RAxML-NG. So basically a short introduction. Sorry, I cannot avoid that because yeah, I mean, also Julia did the introduction, but I also have a short introduction here. So what's actually maximum likelihood inference? We have an input multiple sequence alignment, and somehow we infer a binary tree. And that's the phylogenetic tree inference. So this is a black box actually. And how do we solve this difficult problem? Luckily, we have RAxML-NG, and we don't have to think of that. So RAxML-NG does the maximum likelihood inference that we're interested in. But the problem is that sometimes takes ages to finish. Why? Because it is an NP-hard problem. That's how we characterize it in computer science. So as Julia said, the number of possible topologies grows super exponentially. And there might be some tricky cases where the tree search can actually take for ages to finish. And we have an idea on how we can accelerate that process. We can actually incorporate the Pythia tool into the pipeline of RAxML-NG, so actually to predict the difficulty score and modify the heuristic of RAxML-NG accordingly. And this whole configuration is called the adaptive RAxML-NG. And that's what I'm going to present now. But first of all, some basic steps, because I'm not sure whether everyone is familiar what happens in the background. So what is actually tree inference? We have an MSA, and then we start from an initial tree. What does it mean actually that we assign its taxon to each one of the leaves of the binary tree? And let's say we start from a random tree or a tree with a good parsimony score, but let's say with a random tree. This tree has a likelihood of -100. What does it mean actually? Well, it is a score. It is a probability of between 0 and 1, and we take the log likelihood of that. That's why it is -100. But we don't care about that. We just care to increase that number to make it better, like -50 or something. So what we do just to maximize the score of that, well, I mean, we can do some topological moves. So we have this tree, and let's say we prune these two subtrees, and we interchange them, and somehow the likelihood has been increased. And we do that once more, and again, the likelihood has been increased to -40, and then we optimize the branch lengths. Why that? Because well, the branch lengths and also the model parameters of the evolutionary model contribute to the likelihood score. Well, so these are continuous arguments to this function, and we need to optimize them as well. And now we conclude with a likelihood of -35, let's say. And this is the final tree, and we are happy, we found it. So basically the heuristic is a set of topological moves combined with continuous parameter optimizations. And yeah, the concept of difficulty once more is that for some data sets, all independent maximum likelihood tree searches, what does it mean independent? It means that we start here from different topologies, random topologies, let's say. So all independent maximum likelihood tree searches lead to a single or two topologically similar trees. And we say that in these cases, the dataset expresses a clear phylogenetic signal. So once again, this graph that we start from an MSA on easy datasets, and we conclude to only a single topology, this green tree here. But on difficult datasets, a weird scenario can be observed that independent tree searches yield topologically highly distinct, but yet equally likely trees. And these trees are unfortunately statistically indistinguishable when we evaluate them, for example, with IQ32 significance tests. And we have this graph in this case that we have multiple tree topologies here in the end, and we cannot decide which one actually is the most fitting one. Some other comments is that apart from these two extreme cases that we demonstrated, there is a whole spectrum of in-between datasets, and so with intermediate ones, if we can characterize them as such, and this behavior is essentially quantified by Pythia. So what is Pythia for us? It's a black box, and we actually give an input MSA into that tool, and Pythia produces one number, generates one number between 0 and 1, which corresponds to the difficulty. So we start from easy datasets from 0, and we end up to difficult datasets of with score 1. And the first thing that we can do is empirically classify the dataset. So let's say that easy datasets are considered these datasets which have difficulty at most 0.3. Then from 0.3 to 0.7 are intermediate cases, and then above 0.7 are the difficult datasets. And there is also one recent study which was conducted by Höller et al. Or Dimitri, a colleague of us. And so Dimitri compared RAxML-NG, IQ32, and FAST32 on datasets with varying difficulty. And what he observed is that on easy datasets, all tools perform similarly in terms of likelihood and topological accuracy. On difficult datasets, all tools perform similarly, but only in terms of likelihood score. Because topological comparisons, well, first of all trees inferred were topologically distinct, but second of all, there is no sense in comparing such trees, like there is no clear phylogenetic signal. And he also observed that on intermediate MSAs, RAxML-NG and IQ32 infer significantly better trees, which means actually that superficial heuristics on intermediate MSAs are not recommended to be used. So we need more intense heuristics here. And based on these observations, we have the adaptive RAxML-NG. So what standard RAxML-NG does is that it initiates 10 random and 10 parsimony trees, mean that trees with these are trees with a good parsimony score. And on each one of them, it applies a tree search heuristic by exclusively using SPR moves. What are SPR moves? Well, it's subtree, prune, and regraft move. What is that? We have a prune subtree that we actually prune it, and we regraft it to a different branch. And we evaluate the likelihood score of this topological move. And there is also the concept of prune regraft distance, which in this case is equal to 2. So actually it is how far is the regrafting edge from the pruning edge. And here we just traverse the tree and we see that it's like 2 edges apart. And the SPR round actually is a component of RAxML-NG heuristic. So it's a sequence of SPR moves which follows a hill climbing heuristic. It's a greedy approach. And there are actually 2 types of SPR rounds in adaptive RAxML-NG, but also in the standard RAxML-NG, the fast and slow. And the SPR radius parameter so denotes again the maximum distance in this SPR round. So I will demonstrate one example right now. Let's say we run an SPR round with radius 2. That means actually that RAxML-NG will evaluate all possible topologies with a regrafting distance up to 2. So in this case, we have all these green edges, which are potential regrafting edges. And what RAxML-NG does is that it prunes the subtree and it regrafts it to all these green edges, and it evaluates the score. So what happens in fast SPR round, it just does that and it keeps track of the calculated likelihoods. And in the end, it picks the topology with the best likelihood from this list. And it continues. So what happens in slow SPR rounds? Well, quite the same process, but this time the 3 branches around the insertion point are optimized. So we have a better approximation of the likelihoods, of the true likelihoods of the topologies. And actually, another component of the slow SPR rounds is that the top 20 topologies with the best score are stored in a list. And after the SPR round has finished, RAxML-NG optimizes the branch lengths from these 20 topologies and picks the one that has the best likelihood. So in adaptive heuristic, we modify the number of maximum likelihood researches, and we modify also the radius and fast slow SPR rounds, and we further introduce the NNI moves. So regarding the first 2 components, Here we can see two plots. The x-axis is the difficulty, the y-axis is the number of tree searches. So as we can see here, these bell curves find a peak on the difficulty of 0.5. So when we have difficulty 0.5, RAxML-NG initiates 10 random and 10 maximum parsimony starting trees. Here we can see that the orange curve is for random and blue is for parsimony trees. And when we have, let's say, difficulty 0.2, it conducts, let's say, 2 randoms and 4 parsimony trees. The same thing happens also with SPAround, that when the difficulty is 0.5, it finds a peak of 30, and it starts from 5 for 0 and difficult datasets. And as we can see, actually, is that as we move closer to 0.5 in both cases, the heuristic becomes more extensive. And a last comment here is that we can see a gap here. The maximum parsimony trees curve is wider due to an observation made by Morel et al. So he observed Benoit, so the person who was about to give a talk, but in the end we give a talk for instead of him. So he made the observation that parsimony starting trees lead to significantly better likelihood scores. So basically there is no point in using so many random trees as initial trees. So we give a boost to parsimony trees. And what is NNI moves? NNI moves is that we just pick an intermediate branch, a central branch, and we prune 2 subtrees from each side and we interchange them. So what is a sequence of NNI? The NNI round is a sequence of NNI moves, and for each internal branch E, we evaluate all 3 topologies by optimizing all 3 NNI neighboring topologies by optimizing their 5 central branches, and we pick the one with the best likelihood score. And the NNI moves are generally faster, so we alternate between SPR and NNI rounds in adaptive RAxML-NG, and this alternation leads to higher convergence speed. And on these difficult datasets, we begin with an NNI round because the probability of rapid convergence on those datasets is relatively high. So the results. The experimental setup, we gathered a lot of empirical and simulated datasets of all possible types. And the simulated datasets, we took them from Holler et al. Study, so from Dimitri's study. And we conducted some filtering, and we ended up having 9.5K empirical and 5,000 simulated datasets. And this is the pipeline. So we conduct standard RAxML-NG tree search, adaptive RAxML-NG tree search, and we compare, first of all, the execution times. We compare the likelihood scores of the 2 output trees, the RF distance, and we compare the 2 output trees on the basis of IQ3 significance tests. So first of all, the difficulty score distribution of these datasets is this one. These are density plots for empirical and simulated datasets. We can see an asymmetry here in difficulty score, but this is actually because due to the definition of the difficulty score per se. I provide an explanation to the paper. So basically what Pythia does is that the super difficult datasets classifies the super difficult datasets in this region between 0.7 and 0.9. And only a few datasets are classified with difficulty above 0.9. But this is actually something that we're currently discussing about Pythia. But apart from that, we gathered datasets with varying difficulty. So we compared the log likelihood difference between the 2 output trees of standard and adaptive version and the relative log likelihood metric. So in 98% of the cases, the LD metric, the absolute log likelihood difference is below 2 likelihood units, which means that either the adaptive version is better because it's just we subtract from the log likelihood score of the standard tree the score of the adaptive tree. So it's either better the adaptive tree search or it's worse, but no more than 2 log likelihood units. And in 99% of the cases, the relative log likelihood difference is below 0.1%. These are the statistical tests. What this plot says actually is that 95% of the cases, around 95% of the cases, we have both versions inferred trees which are statistically indistinguishable actually. The RF distance actually of this is the plot that we illustrate the RF distances of between the standard and adaptive RAxML searches, and the orange dots are for simulated datasets. The empirical dots are illustrated by the blue dots. And as we expect, actually, is that as the difficulty decreases, the RF distance is approaching 0, and as the difficulty increases, so it goes to 1, let's say, for more difficult dataset, we can see that the RF distance between the 2 output trees is increasing, but this is kind of expected. And the speedups, this is the distribution of empirical and simulated datasets for 10 difficulty intervals, and we can see that on easy datasets, we can have speedups of let's say an average speedup of 10, but even on intermediate datasets, we can have speedups of around 2 because of the per round speedup that we gain due to the new heuristic. And regarding future work, we want to play a little bit with the heuristic, what we are currently doing right now, because designing the optimal heuristic can be tricky sometimes, and also experiment a little bit with the statistical tests for early termination. And what we consider now is the sequencing error rate and how we can actually take advantage of that in the phylogenetic tree inference pipeline. And availability, so adaptive RAxML-NG is currently available here in my GitHub under branch adaptive, but we want to incorporate it into standard RAxML-NG soon. The manuscript, the paper is published in Molecular Biology and Evolution, and now we have a very simple demo. So how can we invoke the adaptive version? We just need to type ---- adaptive, and then this is the standard command. So everything remains the same when calling with RAxML-NG. I'm going to show it right now in my command line, and the predicted difficulty is printed out directly at the beginning of the execution. So we have adaptive mode predicting difficulty, and we're happy because this dataset is very easy. And the number of starting trees is modified according to the difficulty, and also the SPR radius, as we have demonstrated previously. And now I'm gonna show a quick example on how we can use adaptive RAxML-NG. So we just call the binary file, we type ---- adaptive. Now let's do a sequential version. The MSA is seminar datasets. Okay, let's pick the easy one. This one is protein, so we specify model LG. Okay, let's also type trees. I want to show something with that. And let's say we don't want files, so no files, ---- no files, generate no files. So first of all, what we see here is that the predicted difficulty is 0.04. The trees are the number of independent researches is modified accordingly. There is a warning that the user has specified the number of initial trees in the adaptive tree search, but the purpose of the adaptive tree search is to determine this number of searches. So this input will be ignored. So that's what I did here with specifying the starting trees that I want one random tree here, let's say. And
Anastasis, just to mention, if you can wrap up to make sure there is enough time for questions.
Yeah, I'm sorry. So basically, this is the execution for the easy dataset. But if we execute the non-adaptive mode in this dataset, so we see that the runtime is significantly slower. And I want to show something here with the difficult dataset. So let's say that we go here and pick up the difficult MSA. And we want the GTR plus G model here because it's a DNA dataset. And we call the adaptive mode. What I did wrong. Okay. Yeah. So the difficulty here is 0.85, which is rather difficult, and RAxML-NG prints a warning here that it's hard to analyze datasets and the output is not to be considered as representative for the true tree. So basically, that's how we use the adaptive RAxML-NG. And thank you. And I'm open to questions right now. And I also thank Julia because she contributed to my work a lot.
Thank you, Anastasis. Okay, so we can start with the questions and discussion. So maybe I can start with the questions that are already posted in the chat, and then we can have more. Okay, so from YouTube, there was a question by Bushra Sahid that asks, could Pythia also work on approaches adopting the multi-species coalescent?
So Pythia uses RAxML-NG internally, and as far as I know, RAxML-NG does not implement coalescent models. So I guess, I mean, as long as it's an MSA, your data, you can analyze it with Pythia. The thing is whether it will predict the difficulty that is relevant for your analysis. So what Pythia internally uses as model is for DNA data, just the GTR plus G model, for protein data, the LG plus G model, and for morphological data, the binary model. So in the future, we want to adapt it to let the user set the model. But as long as RAxML-NG doesn't implement coalescent models, I think it's probably not the best method to use here.
Thank you, Julia. So maybe some future work, we'll look into that. By Camila Mazzoni, very interesting tools, thank you. On a sliding window approach, where one infers trees for different windows across a genome alignment, should one remove those windows with high difficulty scores? So in a sense, would you recommend using subsets that suggest it's easier to infer the history of the taxa, or should you just analyze everything?
So if I remember correctly, Benoit once tried to implement a difficulty-guided species tree that is inferred from gene trees. And he noticed that it doesn't really work. So the difficulty isn't really informative in terms of which gene tree better represents or should be weighted more in the species tree. So I don't think you should discard these MSAs entirely or these subsets, but maybe try to increase the window. So try to like incorporate more data for these difficult windows by just expanding the window in both directions. To have like a more conclusive phylogeny for this particular subset of the gene. genome, not gene.
Great, thank you. And one question from Robert Waterhouse. I think you said you tried a few metrics before choosing the 10 for the Pythia model training, the aspects. What other types of metrics did you try? Especially curious if you tried MSA completeness metrics.
So I'm not sure what MSA completeness metrics refers to, but we tried, for example, a tree likeness score. So it's, I think it's called also delta plots, which is based on quarter distances between subsets of taxa in the MSA. But it ended up not contributing that much to the prediction accuracy, but it was very computational intensive to compute, because when you're trying to compute all quartets, that's all calculus n to the power of 4, I think. So it's very compute intensive. And what we also tried was to not only use features based on MSAs, but to actually use features based on a single maximum likelihood tree inference, because we thought that maybe like the branch length statistics could be interesting for the prediction, or the RF distance of the starting tree in RAxML-NG and the tree that we end up with. But again, the prediction accuracy did not improve as much as we'd hoped, and it wasn't worth the runtime cost. So this is mostly the features that we also regarded. I hope this answers the question.
Yeah,
thanks.
Okay, great. Another question by Lada Jovovic-Stank. Hope I said that right. If I use adaptive model when running RAxML-NG, is there anything else I need to change in regards of parameters according to Pythia score once I get the output? Also, does it work on multi-gene alignments for species tree inference? That I think goes back
the
to
second
the
one is this one, multi-gene alignments for species tree inference. Yes, I'm not sure what you mean by species tree inference. I think you mean maximum likelihood inference on multi-partition data, because species tree inference can also be reconciliation or.
Yeah, it might be that maybe Lada can clarify, but it might be also referring to coalescent-based methods.
Okay. Then you just need to run only Pythia to get an insight of the difficulty score, because adaptive option just runs the maximum likelihood inference. But regarding the Pythia parameters, the answer is simple: no, unless you want to add more parsimony trees. So there is an option minus difficulty parsimony trees, so df parsimony trees. I have to check it out. I think it's documented, and if not, I will document it soon. But so actually what Pythia does is that it infers 100 parsimony trees at the beginning. You can modify these parameters and maybe just infer more, like 1000, depending on what you want. But I would suggest not to modify that number. It's not useful to modify that. And apart from that, you can just run your analysis with only using minus adaptive. Yes, multi-partition data. Then you can just use the minus adaptive option as it is normally used in RAxML-NG. Like the input, the rest of the input can be normally the input of standard RAxML-NG.
Great, and sorry for the confusing the name. It's Lada Jovovic. It was just a merging of words before. Great, so maybe I can take also the opportunity to ask a couple of questions. And then we can continue with
it also.
Yeah, okay. So one question I have for Pythia, I'm wondering actually, is so in phylogenetics, the traditional theoretical expectation of what a difficult tree is, it can be divided in two things. One thing is a data set that is uninformative, so it's a short alignment or an alignment that doesn't have enough side patterns, or it can be a data set that reflects an evolutionary scenario where we had very short internal branch lengths and very long external branch lengths. So this means the faster the speciation events happened and the deepest in the past they happened, the more difficult this data set would be. So I understand Pythia, or I assume based on what Julia presented, does understand the difficulty in terms of lack of data, but I'm wondering if it also identifies difficult cases as in difficult evolutionary scenarios. So have you seen Pythia identifying such bush-like phylogenies to be difficult cases just using the alignment?
Actually, we've seen something like this in language data. So we had a master's student that analyzed language data with Pythia, and language data also has the problem that there are some branches that are very short and other branches that are very long. And apparently language data in general is rather difficult, if I remember correctly. So it did identify this problem. And I think it works because of the how we quantify the ground truth difficulty, because it's based a lot on the topological distance between trees. And even if you have a lot of short branches, then you will get a lot of distinct topologies because the branches will be mixed, right? So if the branches are very short, it's not that easy to resolve these branches. So that will contribute to a high topological distance, which is then reflected in the overall quantification of the difficulty. So I would say in high level, yes, it mostly quantifies the information content, but I think it can also detect these events.
Great, yeah. Yeah, so I have a lot of applications then for Pythia to, let's see. And another short question for Anastasis. I'm wondering if by using the adaptive RAxML heuristic, there is a chance of missing out on a tree space that ends up being where the maximum likelihood is, if you observe such instances.
So the answer is yes, but the complete answer is that there is always this chance of missing out some potential peaks, because even the 23 searches that RAxML-NG does are not enough. So yeah, well, I mean, what we do is that on easy datasets, we just conduct fewer tree searches because the probability of inferring the true topology is higher. So, and yeah, basically that's what an experienced user of RAxML-NG would do anyways. The user would decrease the tree inferences by themselves. The number of because the default is 20, so basically the user would do like 5 by themselves. But apart from that, I would also like to add a comment based on what I've seen. So basically, unfortunately there are some topologies that cannot be retrieved by the current data that we have, and some data sets are difficult, and that and basically in that case you have to modify the data set either by increasing the length of the sequences or just filtering out some taxa and hoping that the difficulty becomes the difficulty score decreases. Yeah, apart from that, on difficult data sets, there's always a chance of missing out topologies. But anyways, the signal there is too fuzzy, so we cannot make anything with that. We just conduct some superficial heuristic. So yeah, that's the tricky part actually that I was talking about, that to design an optimal heuristic, and I'm working on that right now. The version which is currently available is usable. Somebody can use it and infer meaningful results, but there is like a lot of potential on how the heuristic can be modified. Yeah. I'm sorry if the answer is a bit confusing, like it confuses us as well. But
no, but it's understood though that it's early days in this development. We didn't have something like this before, so I'm sure, yeah, with time there can be even more improvements for the very difficult cases. So yeah, one last question. Maybe you covered it a little bit, but you can think if you want to add something. By Teresa is a general question. A user can, need two faces. So what do we do? How do we react on a bad Pythia oracle? Do I quit from analysis, analyzing the data, or adapt in a certain way?
We just omit this information from the manuscript. No. But I don't know, maybe Julia has something to add here.
So, I mean, you don't always have the option to just quit the analysis, right? So you might need the actual information. And I'm not a biologist, I'm a computer scientist. So maybe these suggestions don't really make sense or are not practical. But maybe you can remove taxa from your alignment that you already know the evolutionary history for. So maybe it's been recovered before and there are conclusive studies and like studies that actually show the history for this subtree. So you can just leave one representative of this subtree in the MSA and then see if this improves the difficulty. And if you can, maybe you can add more sequence information, which of course is probably not that easy to do. For us as computer scientists, it's easy because we can just, we don't really regard the biological meaning of the data. But
Okay, it turns out that Julia, we cannot see Julia now, right?
Okay, well, I thought I froze, but it was Julia.
No. So, yeah, I mean, more or less what we said is that sometimes you need to add more information. And there is also a quote like sometimes more data is better than better models. So
yeah, well, actually, also my thought on that maybe is I assume that if you get a bad score from Pythia and you can imagine ways of improving the data set, that's a good strategy to go on. Like if you imagine that your sequencing has gone bad or your taxon sampling is really poor, then it might be an indication how to improve that. But if you have done a really good job in designing your study and doing your analysis and you get a very bad score from Pythia, then I guess it's an indication that you need to make much more effort for reconstructing this evolutionary scenario because it might be challenging as a history, like it might involve introgression or incomplete lineage sorting or other sorts of heterogeneities in the evolution, and then just doing a maximum likelihood would not be enough.
Yeah, but I mean, I totally agree with Julia with what she said that we can just leave one taxon as representative for the whole subtree because sometimes we don't know the in-between relationships, but we can know the more abstract structure of the tree, which is also meaningful.
Okay.
I actually wanted to add something. Sorry for the internet issues. So that's modifying the MSA. But if you can't modify the MSA, then it's very important that you don't just report the best tree that you get from your analysis, but to do some summary of all the trees that you get. So you can compute the consensus tree. You can summarize all the trees that are statistically not indistinguishable from each other. But I think bootstrap support might be less helpful because we get low values anyway. But it's very important to not just report a single tree if you know that your data set is difficult.
Great. Yes. Thank you, Julia. And I think with that, we can wrap up. It's more than an hour now. And thank Julia and Anastasis again for giving us these great talks and many things to think about. And yeah, hope to see you again.
We also thank you for the advertisement and for your host here in the online meeting.
Yes,
And
thanks a lot.
Thank you so much.
Thanks, everyone. Bye-bye.
Bye-bye.
Bye.
yeah.
Bye.